All right, in chapter 18.3, we're going to talk about surviving in the host, right? So we got in, we've attached, and now how do we survive in there as a pathogen? We're going to talk a lot about the difference between intracellular pathogens, the ones that live inside of cells, and extracellular pathogens. They're going to do different things because they need to avoid different parts of the immune system. So a lot of this is about immune system avoidance, like I said, and then we'll talk about some very specific strategies that each use. And I would like you to know one strategy that intracellular pathogens use and one that extracellular pathogens use. So, microbes get into the body and they're constantly sensing where they are. So microbes don't have eyes or ears or things like that. The best way to describe it, I think, is they're tasting their environment. They're sensing what molecules and chemicals are around them. Which is essentially like our sense of taste. And they use this to figure out if they're in a host, if they're outside a host. So things like temperature, pH. So when a microbe gets ingested, right, you eat something that has salmonella bacteria on it, it goes into you, you're warm, so it senses the change in temperature, and then it senses the stomach acid when it hits your stomach, and at that point it knows it's inside the host. That's going to lead to it switching on many genes, particularly to avoid the host immune cell. Extracellular pathogens live outside of cells, so they have to avoid a lot more parts of the immune system. They have to avoid antibodies. They have to avoid the innate immune system. They're very specialized at doing this. There's ways they can do that. We'll see. Intracellular pathogens have a bit of an advantage because they can hide inside of the cell, away from the immune system. Although there were mechanisms, right, the cellular immunity that we studied that allow the body to sense when there's a microbe inside of the cell and then to destroy that cell. So we'll look at how microbes can avoid that destruction. We're going to start with the extracellular pathogens. I'm going to list off a few ways that they avoid the immune system, and I'll show a diagram in a moment. One of the big ways that they avoid the immune system is with a polysaccharide capsule, which coats the bacteria. It's extra stuff around the cell wall that is sticky and slimy and can help avoid phagocytosis from binding. The only way to grab onto these microbes is to use the opsonizing antibodies that bind to the capsule, and that gives the phagocytes a little handle to hold onto to engulf them and destroy them. But, as we'll see, there are certain microbes that can turn antibodies around. So normally, the Y part of the antibody binds to the antigen, right? So that sticks to the cell surface, and this FC region is the handle that the phagocyte grabs onto. Well, there are some devious microbes that actually bind the FC region and leave this part sticking out there. That's very clever. Some pathogens just switch up what antigens they make on their surface. So they'll have multiple genes for the same protein, and so they'll make protein A for a while, and once the immune system starts to catch on, they'll switch over to version B of that and make that for a while, and then C, and so on and so forth. That helps them avoid the immune system recognizing their surface antigens. Another one is cell-to-cell communication. We don't think about microbes communicating, but they're constantly talking. And a lot of microbes will invade the cell and they'll start growing, but they don't switch on their genes that cause damage, right? They don't switch on their pathogenesis genes. They wait and wait until their numbers get high enough, right? If you had one bacteria that went in and it started making toxins and things like that, the body would notice that real quick, and all the immune system would rush there and kill that one cell. So what the bacteria do is they come in and they multiply and multiply and multiply until there's hundreds, thousands, millions of bacteria in there. And then all together, they switch on their pathogenesis genes, their virulence factors, and that overwhelms the body. So this can be very, very dangerous at this point. Okay, so let's look at the graphic of this. Once we get in, microbes can make a capsule, right, that helps them be slippery. They can change up their cell surface antigens. They go from making the blue version to the red version. Does the same thing, but slightly different shape, so the immune system no longer recognizes it. My favorite is sequestering antibodies. So they just turn the antibody around and face it the wrong way, and then it can't bind to them. I liken this to, you have someone that has really short arms and they can't reach you, so you put your hand on their forehead and they can't hit you, right? It's kind of the same thing. I think it's very insulting, basically. The bacteria, they take that antibody and just turn it around and it no longer works. Other microbes can secrete fake cytokines, things. They basically trick the immune system. They give it the all clear signal and say, nope, no problem here, nothing wrong here. Or they screw with cells and they cause them to go other places or do other things. So they're just messing with the immune system there. And then we have the auto-inducer system, which is the bacteria talk to each other by this quorum sensing mechanism. And once they get to a high enough level, the auto-inducer gets to a high enough level and they all switch on their virulence factor genes and cause illness. So these are all ways extracellular pathogens avoid or alter the immune system to stay inside and grow. Intracellular pathogens can avoid the innate system and that antibody humoral system because they're inside of cells. The body can't detect them there. So they're hiding a lot of their antigens from the immune system. And this works, but we still have that cell-mediated immunity there. Some pathogens are what we call facultative intracellular pathogens. They can invade host cells, but they also survive and grow outside of the host. Salmonella, Shigella, Listeria, right? Listeria can grow in your fridge, but then it can also grow inside of your cells. There are other ones that are obligate intracellular pathogens. They invade and only reproduce inside of host cells. We talked about the chlamydias as an example of this. Rickettsia, Rocky Mountain spotted fever, I believe. Coxiella, which causes Q fever, common in dairy animals, and Bartonella. All of these are intracellular, obligate intracellular pathogens, so they have to go inside of a host. There's three general ways that intracellular pathogens can avoid the immune system and deal with the cell. Most of them trick their way into cells by inducing phagocytosis. So normally phagocytosis causes microbes to be brought in and destroyed, right? But different ways to avoid that fate here. Number one, there are some microbes that are just so tough, they allow the phagosome to fuse with the lysosome. Remember, the lysosome has all kinds of toxic chemicals and things in it. They let it fuse. They're like, I'm tougher than whatever chemical you can throw at me. So they're totally welcoming of that, and they just live just fine inside of that toxic, nasty phagolysosome right there. So that's one strategy, just deal with it, be tough. Strategy number two, basically, they nope out of the process. They go, okay, I'm in the phagosome, but nope, you're not going to fuse the lysosome with me. They give false signals that prevent that fusion from occurring. Salmonella does this. Salmonella then goes on to go across the cell membrane. Salmonella is even more devious. It gets into phagocytic cells and goes to the lymph nodes and gets distributed throughout the body. So it uses our own cell defenses to its advantage. The final mechanism is to just escape from the phagosome. Shigella and Listeria do this. They break out. And they actually hijack the actin filaments in the cell and use that to move around. And they can actually move from cell to cell in this manner. So let's go and watch an animation of these processes.
A bacterial pathogen capable of hiding in a host cell initially enters by endocytosis. The pathogen induces the host cell to take it up by phagocytosis and ends up in a phagosome. These intracellular pathogens, which live inside host cells as unwanted guests, gain temporary safe harbor because antibodies and phagocytic cells will not penetrate live host cells. Once inside the phagosome, intracellular pathogens have three options to avoid being killed by the fusion of the phagosome with a lysosome full of digestive enzymes. A single species can use only one of the three mechanisms. First, some pathogens can simply escape from the phagosome before fusion occurs. Pathogens such as Shigella and Listeria use this strategy and then enjoy unrestricted growth in the cytoplasm. They form actin tails that push them around the cell and even into adjacent cells, where they continue the infection. These microbes can spread from cell to cell without ever encountering the extracellular environment, where they would be vulnerable to attack. Alternatively, other pathogens, such as Salmonella, escape intracellular death by modifying the phagosomal membrane or other cytoplasmic components, preventing the fusion of the phagosome and lysosome. Salmonella enterica, serovar typhi, the cause of typhoid fever, prevents phagolysosome fusion after it invades intestinal cells. Salmonella exits the intestinal cell and invades awaiting macrophages. Back in a phagosome, the bacterium reproduces, producing a large number of Salmonella bacteria. The macrophage enters regional lymph nodes and infects other cells. From there, macrophages disseminate the bacteria through the circulatory system. A third way of escaping death inside a cell, performed by bacteria such as Coxiella, is simply to tolerate phagolysosome fusion. In this grin and bear it strategy, Coxiella bacteria reproduce in inclusion bodies in the acidic phagolysosome environment.
I particularly like the Salmonella example in there. I think it's particularly devious to use the immune system to help it spread throughout the body. This is just one example of how intricate these processes can be, and they've evolved to be very good at this, which makes disease worrying. Here we can see some actual micrographs of the process happening. Here's Coxiella burnettii, basically living inside a phagolysosome. It's just chilling. It's got all those toxic chemicals in there, but it's perfectly happy. Other ones like Shigella, here you can see it using those actin tails to move around through the cell. Okay, so we're surviving, we're avoiding the immune system. There's a lot of ways this can happen. I would like you to know an example of intracellular and an example of extracellular microbes, how they avoid the immune system in this. In the next section, we'll talk about the damage that microbes can do to the host cell.